Okay, so now we're going to move on with the taste of smell, and perhaps we'll get into hearing also. In this picture, obviously we have a tongue in front of us, and animals need to be able to pick up on taste. It's really important for their ways of survival and evolution and figuring out whether something is edible or not. If it's sweet and a high energy source, they're probably going to want to eat it depending on the type of animal. But a lot of animals are probably going to have taste buds or ability to pick up on things that help them to survive. Sometimes taste buds aren't on tongues, though. It can be found on antennae and things like that, and feet, depending if you're an insect. But we're going to go ahead and look at vertebrate and look at tongues of vertebrates like ourselves that have backbones. And so here we have taste buds that are located in these grooves in tongues, and that's where we'll find these buds of tongue, of hair cells, taste buds. So obviously there's all sorts of little projections of papilla that help to move food around in your mouth, and also you have salivary glands that help to moisten your food and help for the fluids to go down into these crevices to reach the taste buds. So here's the anatomy of the taste bud. You can see that there's a lot of epithelium cells, and then you're going to find areas where they're like little buds of support cells and gustatory cells, also known as taste cells, gustatory cells. And you'll see that there are specialized chemical receptor cells that are adjacent to or synapsing with sensory cells that will then travel to the gustatory portions of the brain. So as you can imagine, either action potentials or some type of chemical synapse will take place that will influence these nerve cells sending information to your brain, and it'll help you to perceive if it's salty or sweet or sour. And so we're going to get into how do the taste buds do that. So some taste cells can pick up on different flavors, but they tend to be predominantly found in certain regions, where like the tip of the tongue is better at picking up on salty flavors. A little bit behind that would be the sweet flavors, on the sides sour flavors, and at the back more bitter flavors. That doesn't mean that you couldn't pick up some of those flavors in the front part of your tongue. But again, these are the areas where you seem to find taste buds that have a higher predominance of those particular types of taste buds and receptors. Why are these flavors particularly important? Well, often if it's a bitter, it might represent that it's a poison that the animal needs to be able to reject quickly and not swallow it. So it'll, potentially barf up the food. If it's sweet, obviously that might be a sign that this is a high energy source and it should be swallowed. Salts are very important, as you know from learning about nerve impulses and muscles, how important salt is in regulating action potentials. And so this is something that's highly sought out by many animals. And so you can understand why it's so important for an animal to be able to pick up on these different flavors. Here is a actual micrograph or electron micrograph picture of taste cell buds. And you can see here's a taste pore, and you got your taste cells. And here you can see the individual cells. You can see a cell here with a nucleus. The whole thing's a nucleus. And do you recall what the little dark center is? That would be a nucleolus. And as you remember, nucleolus makes ribosomes and things like that for RNA, and also so it's important for making ribosomal RNA. Well, anyway, you can see that the support cells are very tightly joined. So the liquid fluid from your saliva can come into here, interact with the hair cells, but not necessarily go down between the cells. So here is one example of how signal transduction is used for taste reception. What is signal transduction? I don't know if you learned this. You should have learned this hopefully in cell biology. But signal transduction is when you take a signal of some sort and transform it into something your body interprets. Takes a chemical stimuli or a mechanical stimuli and then turns it into this chemical pathway or cascade that ultimately results in action potentials traveling to the gustatory portions of your brain, and then you interpret it as salty or sweet or whatever. And so this is kind of the overall image. And then I'm going to show you another one that I really want you to study. And I want you to draw for your answers for your review of this particular lecture. And so what we have here is our support cells, and you'll notice there's tight junctions, and those tight junctions prevent fluids from coming through. And then it looks like little hair cells or microvilli almost. And then a chemical comes along and attaches to the top of these hair cells, and it will trigger a chemical cascade that we call that signal transduction that causes calcium to come in. When do we remember seeing calcium before? When else did we see calcium? Well, obviously we've seen calcium used for muscles, and we've seen calcium is really important for exocytosis in the axon terminal. So we're getting to see the same basic idea. Calcium is flowing in either from probably some type of endoplasmic reticulum thing, most likely, I would guess. Either way, it's a calcium storage vacuole of some sort that's inside the cell. It's not really specified in this picture. Or calcium's coming in extracellularly. It causes a kind of a calcium signal or spark, depending on how you want to think of it. But ultimately, that causes exocytosis, just kind of like what we'd seen in the previous lectures with the axon terminal, where calcium came in after the action potential traveled down, causing the vesicles to attach and release calcium. So what's happening here, as you'll see in the next slides, is there's kind of a depolarization that's occurring inside the taste cell. And then that those neurotransmitters are released. And I don't know which neurotransmitter it is specifically. I'll just say it's acetylcholine. You could look it up and try to figure out what it might be. But what I see in this picture is it's triggering depolarization. And what that means is probably sodium is coming in, increasing the inside of this neuron to be more positive, and then ultimately action potentials travel down to the brain. Now, if something is really salty or really sweet, you can imagine more neurotransmitters being released, causing a stronger generator potential and then a higher frequency of action potentials that ultimately causes more release of neurotransmitters downstream that then reaches the neurons in your brain, and then you pick up, oh, this is really salty. So again, remember your sensations, that while you imagine them or perceive them on your tongue, it's really in here that you're perceiving it. And we know it's here, but it's really here. If that makes any sense. And again, it's all these action potentials. It's depolarization. It's changing the cell, making the cell more positive on the inside. That's causing what ultimately is the mechanism that causes you to perceive it this way. And again, I don't know this is a good or perfect analogy, but think about how sophisticated computer programs are. And we know that the software is written with pluses and zeros. And so we get this marvelous responses from computers. And in a sense, nerve impulses are kind of doing something in that realm that gives you this incredible perception. This next picture, I think, helps you to really, and this is the one I want you to learn, see how the senses, taste of sense works in these taste buds, these gustatory cells. I will need to double check. I can't quite recall, since it's been a while since I've read up on this. But most cells, I believe, are specialized either to be salty cells or sour cells or whatever. Though there may be some overlap in some cells. That might be something that you can specifically look up to double check. But this is showing you all the different ways that taste buds are influenced by chemicals that we respond to, whether it be salt, sour, sweet, or bitter. What about umami? Well, that's another sense that you can also pick up. That is made from monosodium glutamate. If you're wondering what monosodium glutamate is, you can get that kind of flavor. You can buy monosodium glutamate and sprinkle it on your food, and you can make like a corn chip taste like a Dorito by sprinkling this on it. That'll give you an idea of what umami tastes like. It's that kind of Dorito flavor. In addition to the other flavors they put in there, is that kind of little different thing about it that you don't get from when you get some corn chips at your Mexican restaurant. It doesn't taste like Doritos, and it's not just because they put nacho cheese flavoring on it. It's because they've added monosodium glutamate. Or it's like, no, you can make a chip. If I bought some plain old corn chips and put monosodium glutamate on it, it'll taste like a Dorito. So umami is a flavor that's often associated with Asian food and means delicious. So anyway, again, a taste bud may be focused on just one or two of these types of tastes. This diagram is giving you an idea of what all of them do on one particular hair cell. It's like I'm saying, it may just be a hair cell specialized for salts or sour or sweet or whatnot. But this diagram is kind of showing you everything. I believe they're specialized, but I might be wrong. It's been a while since I looked at it, so maybe worth checking out. So let's go over this. So let's imagine this is a salt, a taste bud that picks up on salt flavors predominantly. They're going to have sodium channels. And as you know, sodium flows in. And when sodium flows in, what happens to the inside of a cell? It becomes more positive because it's a positive cation. And so when it becomes more positive, the inside depolarizes, kind of like a generator potential. I think it would be characterized more of an increase. And then calcium will flow in either extracellularly or from intracellular storage. I'm not sure if it's some type of endoplasmic reticulum or not. I would just made that guess because of the sarcoplasmic reticulum in muscles. But either way, the sodium increases, the sodium increases, depolarizing the cell, opening up voltage-gated channels for calcium. Calcium comes in. Calcium triggers exocytosis, and I believe the calcium actually attaches to receptors on the vesicle that causes the vesicle to attach to the cell membrane, and like a bubble, because the vesicle is a bubble holding the neurotransmitters, attaches and opens up, becomes part of the cell membrane, and the neurotransmitters flow through. And again, that's going to cause depolarization of the neuron. Sodium is going to flow in, and then ultimately, if it reaches threshold, we're going to have action potentials traveling to the brain. So that's how salt does it. That's how salt triggers the action potentials that ultimately result in your brain picking up on it. Again, remember, this is signal transduction. We're taking a signal from the environment, changing it in the cell, that message, so it can go to your brain. For the purposes of this particular taste receptors, instead of just referring to it as signal transduction, we're actually going to call it taste transduction. Because as you can imagine, we're taking this environmental cue, salt, and then this is what's going to be perceived in the brain. What about sour? Well, sour is an acid. Where do we find acids? Well, we find them in lemons and limes and oranges and other types of fruits. Acids are made up of hydrogen ions, and it's just a hydrogen atom that's positively charged, and that's what an acid is. It's just a hydrogen ion. And if hydrogen ions come in, instead of sodium ions, and hydrogen ions come in, the inside will become more positive, depolarizing the cell, making it more positive. And if it makes it more positive, calcium-gated channels open, allowing calcium to come in. Calcium attaches to the vesicle, and then calcium's released, or excuse me, neurotransmitters are released, triggering action potentials. If the acid can also attach to receptors that close potassium channels. What happens when you close a potassium channel? Remember, the potassium tends to leak out because of this. What is in charge of making that happen? The sodium-potassium pump. Just like we talked about in the very beginning of nerves, we have sodium-potassium pumps here too. And so the potassium levels increase inside the cell when the potassium channels are closed. And when the potassium channel is closed and the inside becomes more positive, the cell depolarizes. Voltage-gated calcium channels open, calcium comes in and causes the vesicles to release neurotransmitters, become more positive. And it gets a little bit more complex when we talk about these chemicals over here. Now, I believe sour could be doing both at the same time. Umami attaches to probably a G-linked protein of some sort. That's what it says actually over here. Remember, G-linked protein is what they call it when a protein and another little protein is attached, and then they come off, and then it causes usually some type of chemical cascade, meaning there's quite a few chemicals involved, enzymes and so forth. That's what we call this multiple signal transduction pathway. But ultimately what happens is it causes the cell to depolarize, calcium flows in, and then we have vesicles and neurotransmitters being released, action potentials are generated, sent to your brain, and you perceive these other flavors. So both bitter, sweet, and umami do so with specialized receptors for their specialized sugars or specialized bitter components. It's a lock and key mechanism. And so that is how you can use artificial sweeteners. Artificial sweeteners will attach to the receptors that are normally where sugar attaches and trick your brain into thinking you're eating something that's sugary. But instead, it's that receptor being attached to. So again, you can have an artificial sweetener attached to this receptor, trigger this chemical cascade, increase calcium levels, and cause neurotransmitters to be released. Anyway, this all travels to the nerves and so forth. So I think I'm actually going to hold off on hearing and make that a separate lecture. And so this is the drawing that I want you to provide and explain how taste works. So don't just provide the drawing and write exactly what you see on the right. I want you to try to include a little bit of something extra, whether it be something, what you learned from the lecture, from your summary that you're going to write. It's a little bit over half a page, single space. Maybe something that you looked up. I will try to provide a few supporting videos and stuff for you to use for your homework as well. Anyway, I hope this gives you a little bit better appreciation on how taste buds work. You start to see this kind of the same interesting basic scenarios that you get this cell depolarization, neurotransmitters are released, information is then sent to your brain. And obviously the specifics probably change a lot when you get into the deep aspects of it, but this is the general gist that you'll see over and over again. So anyway, that concludes lecture 11. I believe this is what it is. More importantly, to summarize taste and how it works. And please draw this specific drawing that you see up on the screen right now and explain it in your own words. Thank you.